Okay, so welcome to the new Parafrap web seminar series 2024. So we have actually programmed 11 new talks of the period to December 24, which actually covers the Parafrap activities. And today it's a very pleasure to start with Antoine Claessens, who's actually someone who works, who has now a major focus on field studies. But before he will give his talk, I tell a little bit about how he came to his position where he's now in France, in Montpellier. So Antoine actually did his university studies in Belgium, in Liège. And then he moved for a PhD to Edinburgh to work with, I think, outside Antigenic. And this was, I think, with Alex Rowe, where he learned actually part of the pathogenesis of malaria parasites. All done, I think, mostly in vitro studies. And for his postdoc, he moved then a bit more south to the Sanger Center to work with Dominik Kwiatkowski, where he changed a little bit his program. He looked more at genome-wide studies, where he really got interested in population genetics, where he did some seminal work and publication. And I think that inspired him to go actually back to the field, and he received, I think, in 2014 an MRC fellowship to start a small group, part of a consortium, to go to the Gambia, where he actually then started genomic epidemiology again, doing very important work on Plasmodium falciparum, trying to understand what is clinical malaria, what is asymptomatic malaria, and so on and so on. And with this package, he decided to move to University of Montpellier, where he received first an interim staff position, so that gave him stability. With this, he got a small group, an ATIP grant, which is an independent group leader position. And he also is now a Parafrap member. And with this, he started his own laboratory on clinical malaria, again, trying to understand what actually makes asymptomatic and symptomatic malaria. So he's back to the field, and today his talk is actually focusing on avoiding recognition by the host, immune response, and basically he calls this strategies of stealth. That means staying under the radar of the host immune response, and I think his focus is on a group of surface antigens called the VAR genes. And I think with this, I think I gave enough introduction to Antoine. Please go ahead.
Thank you very much, Arthur, for this very kind introduction. And Happy New Year to everyone. Thank you for joining. Before I talk about malaria, I'm just going to tell you about my New Year resolution for 2024. And that is to talk about climate change for just 1 minute at the beginning of every of my talk. Because that's what we should all be worrying about right now. As you probably all know, I hope, is that the two best things you can do right now to reduce your carbon footprint is to not fly, or as little as possible, and to massively reduce your meat consumption. And it's about the second point. I want to give some interesting facts. Actually, I'm going to ask you a question. If we were to compare the total biomass of all humans, all wild mammals, and all livestock that we feed on, so cattle, sheep, pig, and so on, what do you think is the heaviest? Humans, wild mammals, about 6,000 species altogether, or a handful of livestock species? Well, actually, the answer is that overall, 96% of the total biomass is either human or the animals that we feed on. So this is the wild mammals that we all enjoy watching documentaries about. That actually nowadays only represent 4% of the total biomass. And I think this really shows why meat consumption has such a big impact on climate change. It's because we just need so many animals to feed on. That's it. End of the minute. Now back to malaria business. If you've been to a malaria talk before, you've seen these numbers, surely. We all start with these numbers showing how many cases per year, how many deaths from malaria, mainly children under 5 from sub-Saharan Africa. And as you would expect, most of the clinical research on malaria is being performed on these clinical cases. However, if Plasmodium falciparum was presenting this talk, it would say, I think, hold on a second. Here you're showing the exception, not the rule. The rule is that as far as these numbers are high, the rule still is that nowadays on Earth, the vast majority of all falciparum infection are asymptomatic chronic infection. And that's the elephant in the room. And yet, hardly any research is being performed on these asymptomatic infection. And to prove my points, I could show you WHO numbers, but I could also show you real-world data from studies that we started first in Cameroon a couple of years ago with colleagues from the Centre Pasteur du Cameroun. I've been showing that for the last 2 years, so I'll show you new data from last July. I was lucky enough to go to northern Ghana to start a new sample collection. And in both cases, we find ridiculously high number of children being positive for falciparum. And these children were all at school, so they're all asymptomatic, right? They all play football afterwards. And yet the vast majority are infected. And as part of these studies, we treated everyone with antimalarial drugs, and they worked fine, and people are negative. But we monitor them regularly by finger pricks, and within 2 months, the large majority of all of these kids got reinfected again. And yet, I should mention that in both cases, people are already doing a lot to prevent malaria. Some of them at least sleep on the bed nets, or they got access to bed net at least. There is seasonal malaria chemoprophylaxis that is given to all children under 5 years old. The under 5 are relatively protected. There is access to healthcare. So they're really not the worst places to be. And yet, the prevalence of asymptomatic infection is still ridiculously high. So I'm losing faith about malaria elimination in the near future, to be honest. And yet, if you want to be serious about it, you need to target this asymptomatic infection. You can't just do what we've been doing so far, which is to wait for clinical cases to turn up at the local hospital and treat the infection. We need to actively look for the asymptomatic infection and treat them. Now, after these sobering news, in our lab, we like to look at the positive side of things, and is to say that from this asymptomatic chronic infection, we could learn a lot in terms of host pathogen interaction. More specifically, we recruit asymptomatic volunteers, and we do regular blood sampling monthly from the same ongoing infection. And that should allow us to study the host pathogen interaction in vivo in real time. And this is what we started when I was based at the MRC Gambia in West Africa. In the Gambia, the transmission is a bit particular because it's highly seasonal. There is an 8-month-long dry season. And when I say dry, I mean 0 rain for 8 months. And as you would expect, there is hardly any clinical case of malaria during a dry season. And so what we did is we actually collected samples over the 2 years. Well, I'm going to particularly focus on the last 6 months, in which at the beginning of the dry season, December, January, we recruited 42 people who were all positive for Plasmodium falciparum at the beginning of the dry season, and we followed them up monthly for at least 6 months. And here are the results. 3 people became symptomatic quite quickly, so obviously they were treated and they're out of the cohort. 17 people naturally cleared their own infection, typically within a month. Right? But for the 22 other people, for most of them, the infection lasted for at least 6 months. So these are the asymptomatic chronic infection. This is your reservoir for Plasmodium falciparum, and both my colleagues have shown recently that asymptomatic are indeed a reservoir for the transmission of of malaria. And that's what we want to aim to characterize. So all of my talk today will be trying to understand how is Plasmodium falciparum able to establish a chronic asymptomatic infection in some individuals, but not all of them. And we'll do that starting at the population level all the way down to the molecular level and the single cell level. So first, the population level, just briefly, as I said, we recruited people from 4 nearby villages in the eastern part of the Gambia. Here I'm showing the spatial temporal characterization of malaria prevalence. Each green dot, each dot is a household, 3 to 20 people over 2 years. And the color, so the heat map is showing the prevalence. And some households sometimes have got higher prevalence than others. But overall, there is no hotspot for long-lasting infection, which is what we would expect, of course. There is no one side of the village where all the long-lasting infection would be. It's randomly distributed. But I'm just ticking boxes here. So that was for the host. What about the parasites? Well, we also did genotyping of about 400 isolates. And this genotyping outputs a genetic barcode for each parasite isolate. And the link here means that the 2 barcodes are somewhat similar. Right? The 2 isolates are genetically related, siblings or cousins. But overall, the parasite genetic diversity is relatively high. And importantly, of these long-lasting infection, there was nothing specific about it. We didn't identify some falciparum genome that are specific to long-lasting infection. It looks like, for more limited data sets, any parasite genome is able to establish a long-lasting chronic infection. That's just a general overview at the population level. Now, at the individual level, can we identify who is a carrier over the duration of the dry season? Well, the bottom line is that anyone could be a carrier. We didn't find any association with age of the participant or hemoglobin genotype. Perhaps surprisingly, we find no association with antibody levels against antibodies against falciparum antigen. So you might have thought that these fast clearer infection, these people will have high amount of antibodies, and that would explain why the infection didn't last long. But that's not what we think, what we found, sorry. If anything, the slow clearers, so the chronic infection, these people had higher amounts of antibodies against 17 falciparum antigens. So in case you wonder, these antigens are the usual suspects, like the MSP, RH, and EBA families. So when other studies have found the same, that in slow clearing infection, you tend to have actually higher amount of antibodies. I think the explanation is that these people are just more at risk of being infected with falciparum. And so they tend to accumulate antibodies, but most of these antibodies would not be efficacious. They're just markers of infection. So we did find some stuff that correlated with the duration of infection, and that's the initial parastemia. So in December, at the beginning of the dry season, having a high parastemia was making you very, very likely to have a long-lasting infection. And similarly, the multiplicity of infection was associated with the duration of infection. So what I mean by multiplicity of infection is the number of clones or number of parasite genotype that you identify in an infection. So especially in highly endemic places, people get bitten repeatedly, and so they tend to accumulate different strains of falciparum in the blood, right? So that would be a high multiplicity of infection. And again, high multiplicity of infection is associated with long-lasting infection. So the two things the people you want to target, if you want to reduce the malaria reservoir at the start of the dry season, you want to target the people who have high parastemia and high multiplicity of infection. Right, so having identified these people, We can go now to the molecular level, the slide that you all are waiting for right now, and that's antigenic variation. So this little cartoon here is textbook example of how the parasite would evade the immune system for weeks or months or even longer. The theory says that a parasite expresses a surface antigen, and it would regularly switch from one antigen to the next. Okay, and that would force the immune system to develop specific antibodies against each of these antigens. Right? Now, the main family that is thought to mediate antigenic variation is called VAR genes. So I now need to introduce you to the wonderful world of VAR genes. You will find about 60 VAR genes per falciparum genome, each VAR gene being unique. It's unique because each VAR gene is highly polymorphic. And VAR gene encode protein called PFMP1. So PFMP1 is the same as the VAR. VAR is the name of the gene, PFMP1 is the name of the protein. I'm showing here just a very small part of a PFM protein. You see some conserved motif on the edges, but in the middle, it's super polymorphic. Each PFMP1 sequence is unique. And on top of that, each isolate or each strain of falciparum would contain its own repertoire of 60 VAR genes. There are exceptions like VAR2 CSA, but here I'm just giving the big picture. And in nature, you'll find billions of different strains of falciparum, right? So it means that the total number of unique VAR gene sequences in the world is virtually infinite. However, VAR genes undergo mutually exclusive expression. What that means is that at any given time, there is only one type of VAR gene that is transcribed, here the blue VAR, and that will lead to only one type of PFMP1 protein at the surface of the red blood cell. So that's true for at the single cell level, but in your petri dish or in your isolates in vivo, every single individual cell can, in theory, express a different VAR gene. And so often when you do bulk characterization of your transcriptome, you will find multiple VAR genes expressed within your population, but each cell will express only one type. I'm not going to talk much about the function of PFMP1, but just be aware that despite the mind-boggling diversity of the sequence, it is possible to classify VAR genes in 3 main groups, A, B, and C. And multiple studies have associated group A PFM1 expression with cerebral malaria. So what would happen is that this group A PFMP1 would bind to a receptor known as EPCR at the surface of the microvessels, and that could lead to cerebral malaria in the brain. And group B and group C have got a different function. They bind to another receptor called CD36, among many other receptors. But here we'll really focus on the role of antigenic variation for VAR genes. And in vitro, a lot has been shown about how VAR genes switch from one VAR gene to the next. But in vivo, there is hardly any data that is showing this nice little cartoon here. And the reason is because it's hard. Because these asymptomatic infections, they've got very low parasitemia, so very little DNA or RNA to work with. And so there's plenty of questions that remain to be addressed. Do we actually observe regular switching of VAR genes in vivo? And if so, at what rate? And does the immune system actually produce these specific antibodies over time? And so I'm going to focus on VAR gene expression from these long-lasting chronic infections. So how do we do that? Well, multiple approaches. First, the bulk RNA-seq from an isolate. So it's a freshly thawed isolate. On purpose, we don't culture it, right? We want to have the transcriptome as quick as possible after the parasite was isolated from a human being, frozen in glycerolide, and then we thaw it, We do cell sorting to try to increase the parastemia. And right away, when it's still ring stage parasites, we sequence the transcriptome. Now, we were hoping to do de novo assembly of these vargines, but the data quality is not good enough to do de novo assembly. So why do we need de novo assembly? Well, usually when you do whole genome sequencing, whether it's or whole transcriptome sequencing, you map your reads against a reference genome. In the case of falciparum, the reference genome is called 3D7. However, 3D7, we have its own 60 var genes, right? And no other strain in the world, we have exactly the same vargine as 3D7. So there is no point trying to map your reads against 3D7 if you're interested in two vargines. What you need to do is, well, the different approaches, which I'm showing here, and one approach is to do de novo assembly of all the reads and try to reconstruct these vargines. The problem is that with Illumina sequencing, your read is like 100, 150 base pair long. So it's very short compared to a vargine that's like 3 kb. So you can do the de novo assembly works better on DNA because we can recover more DNA, and so it's better quality sample basically, and that allows you to some extent, based on an algorithm developed by Thomas Otto and others, we can assemble vargine from Illumina. And then what you do is that you take your RNA-seq reads and you map them on your de novo assembly of vargines, and you get some vargine expression. But the best approach, well, the one that is giving us best results, if possible, is long-read sequencing. Because with long-read sequencing, you can produce your very own reference genome for your own isolates. And the problem with long-read sequencing, like PacBio, is that you need much more DNA. So first, from your frozen isolates, you need to culture adapt it for a few weeks. And our success rate is about 50%. So that also means that we lose 50% of our isolates. But if the isolate is growing well in vitro, then that's enough DNA to do PacBio sequencing with collaborators at the Sanger Institute. And then you get a beautiful full-length genome with 14 chromosomes, and you get full-length vargines. And then you can map your reads, your vargine reads, onto the PacBio. So we've got about 45 of these PacBio reference genomes now, but we're only just starting with the data analysis. So I'm going to focus on yet another method, which is a bit old-fashioned, really. But as you will see, it's giving us very interesting results. So PCR-based methods using primers that binds to the DBL alpha domains. I need to give you a bit more background about vargines here. This is a typical vargine, and apart from one or two exceptions, all vargines start with the DBL alpha domain. And this DBL alpha domain is two conserved motifs on the edges, and in between is super highly polymorphic, which makes every single DBL alpha more or less unique. So it acts as a barcode, really. And then in collaboration with Thomas Larsen in Copenhagen, we can sequence with Illumina sequencing this amplicon. And I will give you your vargine expression because each DBL alpha will be linked to its own vargine. And what we do next, once we've got the DBL alpha sequence, is we use this Varia software, which is basically a database of over 100,000 full-length vargines that were assembled by Thomas Otto and Thomas Larsen. And that this way we can predict what the rest of the vargine is going to be from the DBL alpha sequence onward. But I'm going to really focus on DBL alpha from now on. Before I give you the big picture, I'll give you just one specific example, just so you can get an idea of what I mean by vargine expression. So here I'm showing a volunteer, 11 years old male, who was infected over 6 months. Very importantly, it's a monoclonal infection with the same falciparum genotype throughout, right? So we found the same genome at each month. So we know it's the same infection that's ongoing for that period of time. At month 1, we detect basically one vargine and then two other vargines background level, let's say. So we call this a dominant vargine. And you see that every month there's going to be a lot of switching, right? It's another vargine here, another vargine at month 3. But again, in these 3 months, we only find dominant vargines plus some backgrounds. And then at month 4, you find 3 dominant vargines. And then an interesting exception is at month 6 here, you find this dominant vargine that came back. It was expressed right here. This tag 59 was expressed at month 4, switched away at month 5, or was selected against, and then it came back. It came back 2 months later. So that goes against my little oversimplified cartoon here, because this cartoon does not account for what I know called recurrent vargine. So what's happening here? I mean, at least I don't know exactly, of course. I can only make hypothesis. Is that either this particular vargine was not really immunogenic, and so it came back 2 months later, but then you wonder why did it switch away? Or immunity is such short-lived that there was antibodies at the end of month 4 against this particular PFMP1, but then somehow the antibodies were not efficacious enough, and the same vargine came back at month 6. Now I'm showing you another example here from another individual infected with the same strain for 6 months, and again here we find 1 recurrent vargine. So these recurrent vargines are the exception, right? The rule is that most vargines are dominant only once, right? Most of them are detected only once over the 6 months. So there is overall a lot of switching, exactly as predicted. But once in a while, you find these recurrent vargines that somehow came back. I show you a third example for a good reason. So again, you know, similar picture, very few vargines, a few dominant vargines expressed, and occasionally some recurrent vargine. And this one is particularly interesting because we realize that it's the same strain that infected both DC01 and DC04. And as it turns out, these 2 individuals, they're brother and sisters, they live in the same house. So somehow they got infected both with the same strain. I don't know who got infected first, but this acts as in a biological replicate, because we've got the same strain infected 2 different people. And in the first one, you will find 8 unique dominant vargines detected over the 6 months. In the second one, you've got 6 of them. But interestingly, you have 5 dominant vargines that are in common to these 2 individuals. And so this number 5 is much more than you would expect by chance. And so I'm starting to wonder, maybe there is something specific about these dominant vargines. Maybe they're particularly good, particularly useful in the case of a chronic infection during a dry season. Now, unfortunately, as interesting as this sounds, I can't go any further because it was just a chance finding, right? What are the odds of finding 2 people infected with the same strain? But it's definitely something I want to keep in mind for the future. And let me give you now the bigger picture. And for that, to measure the vargine expression heterogeneity, we use a metric called entropy, which basically means that a low entropy is a low diversity of vargine expressed. High entropy means lots of different vargine expressed. The overall distribution is normal. But very importantly, this var entropy is not correlated with parastemia. So there was a risk that in the samples in which we find a low amount of vargines detected, that this would have been a low parastemia, and that's why we don't detect all the vargines. But that's not what the case, because there is no correlation with parastemia. So clearly, our method is sensitive enough to detect all or the vast majority of vargines that are transcribed. And so we can then now analyze this var entropy, just keeping in mind that the entropy is on a logarithmic scale, and on the x-axis are the 6 monthly time points. So you see that the viral entropy tends to go up and down in every sample, so it's quite dynamic. And yet, overall, over the entire population of these 12 people here, the viral entropy is relatively stable. However, here I should mention that there is one, maybe the biggest weakness of our approach is that we don't know when these people got bitten by mosquitoes. So month 1 is not necessarily the beginning of the infection. Some people were bitten a few days ago, some people weeks ago, or even months. And unfortunately, I cannot tell that. So based on the literature, specifically the control human malaria and infection studies, my feeling is that for most of these people, entropy would have been very high in the first few days because the parasite is expressing a lot of different genes, but then quite quickly, the immune pressure would select for only a handful of dominant gene. That's what we see. Another fact, sorry. I thought that in polyclonal infection, we would find more genes expressed. So polyclonal infection means that 2-3 clones means 120 or 180 potential genes. So you should find more genes expressed, right? And it's not the case. So surprising, but my interpretation so far is that maybe the immune systems would only tolerate a very limited number of PFAP1 at a time, irrespective of the number of circulating strains. And so, what does this all mean? What have we found so far? Well, first of all, that at each time point, there is a relatively few number of genes that are expressed at a high level, at least. Very few dominant genes. And quite a few other genes at the background level. So this could be what is called a bed hedging strategy. It's basically the parasites switching to a few genes and testing its host, see whether these particular parasites would not be recognized by the immune system and then amplify massively. In terms of the ABC grouping, I didn't show that data, but basically there is no association with the ABC group. We find just as many A genes as you would by chance. And we do show, yes, that indeed there is gene switching in vivo, plenty of switching, but once in a while you identify recurrent genes. So obviously that brings up plenty of questions for the future. And the most obvious one, but it's super hard to answer, is does each PFAP1 elicit an antibody response, right? The second part of that graph here. But we can discuss at the end, but it's tricky to find a good method to do that. Of course, we want to know what's going on with this recurrent gene. And clearly, sampling people once a month is not enough. We see plenty of switching, but I'm sure that if we had sampled more often, we would have found even more switching. So all we need is to find volunteers who accept to give us blood every 2 days for 6 months. If you know someone, let me know. Right, so now moving on to the fourth part, which is about single-cell genomic. Now I've got a confession to make to all of you. I said that my motivation was to eliminate malaria and asymptomatic infection. It's true, but I also have a second motivation, and that's to identify chimeric gene in vivo. So I need to give you a bit of background here first. So over 10 years ago, with Will Hamilton and the Sanger Institute, we produced clone trees, so basically culture parasites in vitro, did whole genome sequencing regularly, and we realized that the parasite would often undergo ectopic recombination. So by ectopic recombination, I mean recombination between two different chromosomes. And as it happens, a lot of these ectopic recombination would be between two genes. And when that happens, then you generate a new gene, we call it chimeric gene. In this case, it starts with the red sequence and then the blue sequence. This particular gene here did not exist at the beginning of your experiment, right? So this was all done in vitro. You started with your 60 genes, and then a few days or a few weeks later, you've generated a new sequence that did not exist earlier. And we could measure very approximatively the amount of virgin recombination that generated. And it's 1 in 500 parasites will generate one chimeric virgin every 48 hours. So from our in vivo chronic infection, we expect roughly that 1 in 2 parasites will have acquired at least one chimeric virgin, but also SNP and microADLs and other mutations, of course. And so to test this hypothesis, we collaborated with Ian Chisman in Texas, and he's the one who did the whole genome sequencing of hundreds of genomes we've got now. But I'm only going to focus on a single infection. Again, this individual DC05, for which we've got 41 single-cell genome at month 1 and 26 genomes at month 6. So the big picture is, of course, that all of these genomes are identical, right? Which is what you expect because it's a monoclonal infection. We already knew it was the same genotype throughout. And the single-cell data really confirms that. Now, as I mentioned earlier, you cannot map your reads against 37 when you are interested in virgins. You need to do de novo assembly of all these virgins. And sure enough, Marc-Antoine, who was a PhD student, found about 55 virgins for each single-cell genome. And at month 6, there were only 53 virgins typically in each cell. So the same virgins, but 2 of them are missing. I'll talk about that more later. But so far, to cut a very long story short, we have not identified any chimeric virgin yet. And I think I know why, and that's what I'm going to present in the next slide. So what we also did is to look for change in allele frequency over time. Basically, we look for de novo mutations that would have occurred during these 6 months. So there's de novo mutation that occur all the time. There's no doubt about that. But the question here is, are there mutations that increase significantly in frequency over time? And so our cutoff was that, are there any SNP or single nucleotide polymorphism that increase in frequency by at least 0.5, right? Something that was rare or non-existent, and more than half of the cells would bear that SNP afterwards. And with that cutoff, we identified 3 mutations. 2 in green and blue on top of each other. These 2 mutations are non-coding, and 1 in red is a coding mutation in a gene called a perforin-like gene. It's involved in egress. But that specific mutation has not been described in the literature. But the main point here is that something really important is happening here. Something really important is happening because these mutations were barely detectable at month 1 are now represent more than half of all cells circulating at month 6. So what's happening here? Well, first I can redraw my cartoon here with the mutant cells. I'm going to label them as with the red, green, and blue triangles. Let's call it the green and blue population, the ones that bear the mutations. And as it turns out, all of these genomes are also genomes with only 53 virgins, as opposed to the wild-type parasites, which had 55 virgins and didn't have the blue and green mutations. So all parasites were here wild-type at the beginning, but by month 6, these wild-type are a minority of the total population. So what is happening? Well, there are multiple hypotheses at this stage. First is that these 3 SNPs somehow confer growth advantage, and they're slowly but surely replacing the whole population. And in vitro, we've got example of that happening. It could happen, but it could also be that these 3 SNPs have got no function whatsoever, but it's something else that's driving something else that's driving the selection. For example, the blue and green population is expressing a particularly useful virgin, And so these mutations are just hitching a ride with their genome. Another alternative is that there has been somehow, at some point, a population bottleneck. And by this, I mean the population completely crashed. And we already know that parasite density in vivo, it goes up and down all the time, right? Sometimes it goes so far down that it's below PCR detection level, and then it comes back up. So it's plausible that there were, at some point, only a few parasites that survived for whatever reason, and just completely randomly by chance, the green and blue parasites are the ones that survived, and now they're the ones that are thriving at month 6. And either way, whatever is happening, I mean, clearly it's much more dynamic, the whole thing, than I would have thought from a monoclonal infection. It's very dynamic, but it also means that this population crash that occurred at some point has reduced the overall population diversity within that host. And so that would explain why we have not been able to detect any chimeric VAR genes, is because any chimeric VAR genes on the white cells here would have disappeared with the crash. So yes, the hunt for chimeric VAR gene is still ongoing, because we just received, like literally this morning, another batch of 150 genomes. And so watch this space. So let's recap, well, everything, what we've learned so far about asymptomatic chronic infection. First, at the population level, it's quite clear that anyone, well, any semi-immune individual is at risk of carrying chronic asymptomatic falciparum infection. Overall, in terms of the parasite population, or the parasite genetic population, the genetic diversity is still very high, even in the context of pre-elimination in the Gambia. I didn't talk about that, but just something we noticed is that isolates from the same household were 3 times more likely to be genetically related than those from elsewhere within the village. So that makes sense, right? If you sleep under the same bed net as someone else, there's a chance that the mosquito will bite both of you, or you will transmit one to the other. We know that hyperuricemia and high multiplicity of infection at the beginning of the dry season is a clear sign that the infection will last over the entirety of the dry season. And does antigenic variation explain chronic infection? Well, it does look like it, or at least we have shown here that clearly VAR genes do switch at a very high rate in vivo, as we expected. But unexpectedly, we identify sometimes recurrent VAR genes. And finally, at a single-cell genomic level, we have not been able to identify chimeric VAR genes yet. However, it's clear that even within monoclonal infection, these genomes are much more dynamic. There's much more interesting stuff going on than one would expect. And earlier, I was saying that it would be nice to have much more regular blood samples and monthly samples. Actually, we do have that, but in a different context, in the context of a controlled human malarial infection. I don't have time to describe the whole study here, but if you're interested, it's all on medRxiv. Basically, it's another study that was done in the Gambia, where people were infected with 3D7, including semi-immune people, and we got 2 time points per person. And what we show in that paper is that there was a massive amount of VAR gene switching, even within just a few days. In fact, for the one individual for which the 2 time points are 6 days apart, so just 3 generations apart, with a complete switch of VAR gene expression within just 6 days. Of course, it's a mixture between intrinsic switching and selection against specific VAR gene. So yeah, go and read the paper. And last but not least, I would like to thank a lot of people, because it's really every project is a collaborative project. Starting with colleagues at the MRC Gambia, where we collected the samples. With Tön Bosma and Umberto D'Alessandro. For colleagues at the Sanger Institute for the PacBio and and other whole genome sequencing. And particularly, I would like to thank Ian Cheeseman in Texas and Thomas Lafson in Copenhagen. And very importantly, everyone who did the work, because I didn't do anything of what I presented today, basically. So my team who did the work actually is Camille, Prince, Marc-Antoine, Ballotin, Inayat, Mathieu, and Sophia. Thank you very much for listening.
Okay. Yeah, thank you very much. This is fantastic study because we all know how difficult it is to work in field studies, especially when it comes to VAR genes. So we have at least 10 minutes time for discussions. Let me see. Please raise your hand. I have. Okay, I have Olivier Sylvie. Let me see. He asked, he has a naive question. Diagnosis of asymptomatic infection is based on qPCR in your studies with 85 to 96% prevalence, which is impressive. Is it possible that some of this signal comes from prolonged circulation of parasite DNA rather than real infection?
Excellent question. And the good news is I've got the answer. Indeed, well, as we all know, as we all learn with COVID, is that even after treatment, you still have floating DNA in your blood, right? And so I don't have the data to hand here, but basically, thanks to the weekly sampling that we did afterwards, I can tell you that when we give a treatment to everyone, a week later, I would say about a third of the children were still positive, but all of them would have been at much lower parastemia. So that's clearly just parasite DNA that's still floating in the blood. And it takes almost 3 weeks. It takes almost 3 weeks for everyone to be negative.
Okay.
Yeah.
Great. I have still a question, a puzzling question about your recurrent VAR gene that pops up again. And since you only have the DBL alpha sequence, can you actually exclude that this is again a chimeric VAR gene that now has a different antigenic setup, and that's the reason why it actually can come back?
No, that's something we want to test. So what we're going to do now to test exactly this hypothesis is using the PacBio genome to map the DBL alpha reads against the PacBio genome and get the full length VAR gene. Now the full story, the issue we're having is that we don't always have the right PacBio genome at the right time.
Okay.
And
okay, so future will show. I mean, I have a couple of other questions. One comes from Catherine Lavazek. She asked, do you have any data about gametocyte carriage in asymptomatic infections?
Yes, and that's in the Collins et al. Paper from 2022 in JID. So basically, all of these people were gametocyte positive, but like at very low level. And so we got the monthly data, and the gametocyte level monthly was pretty much similar over time.
Okay. And then Derek Robinson, he asked, what was the immune status of the siblings who had recurrent VAR genes? Could they be immune compromised?
Yes. I should, it's a good point. I haven't checked. So we've got these, we've got antibody levels against 17 antigen, but not PFA and P1 in December. We should have them against these two. I have not compared these two. Could they be immunocompromised? I doubt it. I mean, there might be a co-infection, of course, but the HIV rate in the Gambia is relatively low, like 1 or 2% of the total population. So it's probably not an issue.
Okay. Then maybe I can ask